So today we will learn regarding the cytoskeleton. So cytoskeleton is something which will provide the structural support to the cells. So as you can see it on the screen, so cytoskeleton, the basic definition of the same has been mentioned there, providing structural support to the cell. Okay, and as well as cytoskeleton also functions in cell motility and regulation. Okay, so basically we will not go in detail regarding what is the definition of cytoskeleton or what are the basic things to be learned there, but directly we will jump onto the types of cytoskeleton and what actually their function is in the cell regulation and cell motility. Okay, let's go. Basically, there are uh, three type of cytoskeleton structures. You can see, as it is mentioned on the screen, you can see there are three main type of cytoskeletal structures, which are also called as cytoskeletal fibers. Okay, those are microtubules, microfilaments, and intermediate filaments. Okay, so and the basic difference between these three can be seen in a very easily can be understood in a very uh, what we call easy manner. Easy manner. Okay, so the basic difference between three type of cytoskeletal structure is their structure, their function, and their size as well. So the all the things have been compared in this particular table. You can see there very easily. Structure has been compared where the largest type of cytoskeletal structures are nothing but the microtubules, and the intermediate, as the name goes, are the intermediate filaments, and the smallest type of microtubules are nothing but the microfilaments. Those are also called as actin filaments. Okay, then the structure, how does the structure differs? As you can see it on the screen, the structure in microtubules, it will be consisting of hollow tubes, 13 to 14 columns of tubulin molecules. Tubulin are protein monomers. Okay, whereas in microfilaments, it is nothing but two intertwined strands of actin filaments. Okay, then in the intermediate filaments, fibrous proteins supercoiled into thicker cables. And in the bottom of the this particular slide, you can see the different photographs as well, which has been photographed in fluorescent microscopy in different colors. Okay, you can see how what is the basic difference between that and as well as the animated structure of the cytoskeletal structures has been also elaborated there. Okay, you can see the difference here in the microtubules. It is nothing but the tubulin dimer. In the microfilaments, it is nothing but actin tubules which are intertwined together with each other as similarly as that of the DNA filaments will be inter what we call winded or tangled with each other. Okay, then what is the main function? Okay, what is actually the protein supplements which are found in the three type of cytoskeletal structures? Okay, tubulin, it will be consisting of alpha tubulin and beta tubulin. The microfilaments, only actin protein filaments are present. No other proteins are found there. And in the intermediate filaments, the proteins related or proteins falling in the keratin protein family are found. Okay, then the main function, what is the main function of the three what we call three cytoskeletal structures? Okay, microtubules, the largest one of them, will be involved in maintenance of cell shape, that is compression resisting. Will be acting as girders. That means as long as the cell is undergoing compression, that will be taken hold with the help of microtubules. Okay, as well as in cell motility, where the cilia and flagellar movement are controlled by this particular thing. Chromosomal movement in cell division, you are very well aware about that particular thing in the form of spindle fibers. Those are nothing but a form of cytoskeletal structures, more specifically microtubules. Okay, then organelle movements. We'll discuss about this thing in the coming slides. Okay, then what the function of microfilaments? They maintain the cell shape by tension bearing elements. Microtubules, they are involved in compression resistance, whereas microfilaments are involving tension-bearing elements. Okay, then changes in cell shape, muscle contraction, cytoplasmic streaming, which is also called as cyclosis. These all things are undertaken by microfilaments. Then the cell division, that is cleavage furrow formation. Cytokinesis followed by karyokinesis followed by cytokinesis. The cytokinesis will be done with the help of microfilaments. Microfilaments preferably. Okay. And in intermediate filaments, so what actually will be having the function is maintenance of cell. There are also tension bearing elements and anchorage of nucleus and certain other organelles which will be undertaken by intermediate filaments. Okay, then formation of nuclear lamina. That means the nuclear envelope that will also be undertaken by the intermediate filaments. That is how the basic difference between the three type of cytoskeletal structures you can see there. Okay, individually the same thing is mentioned here. You can see, and the thing which I was talking about is the organelle movement. It will be taken with the help of microtubules. Okay, thickest are hollow rods about 25 microns in diameter, and as might be known, they will be consisting of tubulin diameters, alpha tubulin and beta tubulin. So what they will be doing, they are involved in the movement of chromosomes, and another function is tracks that guide motor proteins carrying organelles to their destination. Okay, you can see it in the photograph there. Here, this is the microtubule of one of that type of cytoskeleton, and above it, the motor protein is rested, which is ATP powered. That means adenosine triphosphate. It will be supplying the energy in the cell, and this is the organelle upon which the organelle will be binded with the help of a receptor, and movement will be done. That means the movement of cell organelles within the cell, it will be done with the help of cytoskeletal structures. Good example is microtubules. Okay, so what actually you can compare it with the railway and train model. So the cytoskeletal structure is nothing but the railway, and the organelle and the motor protein which is sitting over it can be compared with that of a moving train. Okay, so the same system is involved in the movement of organelles within a single cell. Okay, in many cells, microtubules grow out from a centrosome near the nucleus. These microtubules resist compression to the cell. This is also a very important function you can see there. Okay, further continuing the same thing, what actually we are trying to tell here, you can see it in the picture depicted here at the slide, right side of the slide, a cell out of which the centriole pair will be is elaborated, which is also called as centrosome, and in that how at actually 90 degrees centrioles are tightly packed with each other. Okay, then if you take a cross section of the same, and if you just with the help of electron microscope, and if you take a photograph of the same, the things are visible in this particular format. Okay, so what actually they are telling, in animal cells, the centrosome has a pair of centrioles, each with 9 triplet of microtubules arranged in a ring. So this is a 9+2 model called as 9 triplets. That means in one particular triplet there are 3 tubulin filaments. Such 9 filaments are clubbed with one another to form a single centriole. Such 2 centriole will make one centrosome. Okay, which replicate during the cell division. Okay, and microtubules are the central structure supports in cilia and flagella. Okay, so you can see the electron micrograph photograph of the villi or what you know, cilia and flagella. They look like this particularly. So they show the wavy movement. So that will be done with the help of microtubules which are present in these structures. Okay, so cilia and flagella, about that they are telling both can move unicellular and small multicellular, both can move unicellular and small multicellular organisms by propelling water past the organism. If these structures are anchored in a large structure, they move fluid over a surface, which is seen in fallopian tube in higher animals, as well as in the intestine, as well as the places whereas the cilia are found in the form of the cytoplasmic extensions. For example, cilia sweep mucus carrying trapped debris from the lungs. Very good example. Okay. Cilia usually occur in large number on the cell surface. They are about 0.25 microns in diameter and 2 to 20 microns in their length. Okay, and they are usually just one or a few flagella per cell. Flagella are same width as cilia but 10 to 200 microns long. That means cilia are small in length and more in number, whereas flagella are lengthier but one or two in number. But their microscopic structure is exactly the same. Okay, fine, thank you.